I'm just giving a talk mainly on the stress-induced infections and disease development in Nile Tilapia, Oriochromis niloticus, and the Catfish, Clarias, Garipinus, and Hawassa Lake, Ethiopia. I'm just concentrating on this mainly because, yes, these two species are very important as far as the resources of Ethiopia is concerned, and most of the people, they depend on this particular species for their consumption. So I thought I can just do some work on this particular one so that how best we'll be able to make a further progress in this direction. So my presentation outline is as follows. I'll be just giving some information with regard to introduction. There I'll be just giving the importance of this particular study. And then I will be just going through the material and methods, mainly giving information on description of the study area and the stress factors, concentrating mainly on the water quality parameters. And then the factors that are just affecting the fish populations, I'm just giving mainly the isolation, identification of bacterial pathogens, and also the collection, preservation, and identification of the metazoan parasites. And then finally, I will be just going for the infestation details with regard to these two species, mainly Nile Tilapia as well as the African catfish. And then some studies have been just carried out on the experimental infection with a major type of the bacterial pathogen, and then the antibiogram, and also the effects mainly on hematology and histopathology, and then the results. And I'll be just giving some information with regard to the conclusions and recommendations. So with regard to the introduction, as you know that fisheries and aquaculture play a major role, mainly for the solving the food insecurity issues, especially in developing countries. Considering this, rather is conscious of this fact, countries all over the world are designing and developing new strategies to increase food production through culture and capture fisheries. As far as Ethiopia is concerned, it is a landlocked country. We are just having the freshwater fisheries, and the freshwater fisheries provide livelihood status for million rural people living in this country. However, of late, the capture fisheries production is declining. You know that once we are just going to concentrate mainly on capture fisheries, if there is a decline of this one, and then there will be food insecurity issues will be there. So these capture fisheries production is declining mainly because of the environmental degradation. And these factors negatively impact mainly the water quality parameters, fish health, fisheries of many of the Rift Valley lakes of Ethiopia. The Lake Hawassa is subjected to intense pollution, mainly by a number of man-induced factors like wetland cultivation, cattle grazing, industrial establishments, recreation, etc. It's mainly because Hawassa is just a developing area in Ethiopia. And because of this one, there is a large number of industrial establishments are coming up. The human population settlement is also increasing. And this is actually creating some problem for the water body, mainly the Lake Hawassa. And apart from that, there is a rapid spread of aquatic weeds in the lake. That is mainly because of the inflow of the nutrients into the lake, which help in the production of aquatic weeds. And that is also a great concern as far as the lake is concerned. And these factors alter the physicochemical and biological factors in the water body. And as a result, there is a stress to the fish. And once the fish is stressed, and it becomes a contributing factor for the infection by diverse groups of parasites and pathogens. Let me briefly tell something about the Hawassa Lake. Hawassa Lake is one of the most prominent freshwater lake in the central Ethiopian Rift Valley. Actually, it is located some 270 kilometers south of the capital city, Addis Ababa. And it is the smallest of all major lakes. It's about 90 kilometers square. And it lies on altitude about 1,680 meters above mean sea level. The maximum and minimum depth is about 21.6 meter and 11 meter respectively. It is a terminal lake, so to say, it is a closed ecosystem, and there is no outflow of water from this one. So once if it is a closed system, then imagine how exactly it is just going to be stressed when there is inflow of pollutants into this environment. The minimal annual rainfall is about 1028. That means there is copious rainfall supply is there. And as far as the temperature is concerned, it is 9 degrees to 29 degrees centigrade. And it's a very favorable climatic conditions for living. And as far as the temperature is concerned, it is 19.7, the average. And this is the Hawassa Lake. And you can just see that this is the northern side, and then this is the southern region. And it is very close to the main city, the Hawassa. And Hawassa, the industrial developments are coming, human inhabitants are there. As a result, there is a lot of inflow into this lake, and which is actually causing stress to the lake. And as far as water quality characters are concerned, water quality samples were collected from 5 stations. Physicochemical characters and nutrients were estimated by following the standard procedures, both on-site as well as in the laboratory that we just did it. And as far as the collection identification of bacteria is concerned, bacteria were isolated from the body surface, gills, liver, intestine, and kidney. And preparation of serial dilution was just made based on the method given by Aloja et al. 2018. And then the biochemical and then morphological identifications were done by the standard methods as described by Holt, Scott, and Hammerich et al. And collection of parasites, especially the metazoan parasites, collected fishes were brought to the laboratory for examination. Mainly the body surface, fins, eyes, nostrils were examined, initially by magnifying lens. Later, we'll be just taking the scrapings and it will be just observed under steam series 2 binocular microscope. And gills and other all internal organs were placed separately and parasites were collected separately and then given, placed in separate media to see that they will be used for further identification. And parasites were identified based on the key for the identification of parasites, mainly by Natarajan et al. 1980, Paperna, and then Eshetima Rahmat Nagdik in Ethiopia. And the infestation was just calculated based on the report of Margolis 1962. And the prevalence is a total number of fish infested by total number of fish examined. The intensity is the total number of parasites collected from total number of fish infested. Then we also did some work on the experimental infection to see that how exactly the major bacterial pathogen is causing problems to the fish. A total of 30 Oriochromis niloticus with a mean weight of 30 grams and length of 14 centimeters were considered. And fish were collected from the Hawassa Research Breeding Center. And actually the seeds were obtained through the induced breeding. And after acclimatization for 7 days, they were grouped into 2 groups. One is injected and the other one is non-injected as control. And fish were intramuscularly injected with 0.1 ml of bacterial suspension containing 1 into 10 to the power of 6 per ml. Then fish were fed with artificial diets at 33% of the body weight. And the gross external symptoms developed, if any, were registered. Also, we did some work on the antibiotic sensitivity test to find out how exactly the antibiotics play a role in controlling the infections. It was done by using standard antibiotic discs. 7 different discs of antibiotics were used: ampicillin, tetracycline, gentamicin, amoxicillin, streptomycin, kanamycin, and erythromycin. The inoculum were prepared with 5 ml tryptophan soil and test tube, and the concentration bacterial cells was adjusted to 5. Then Macfarlane's standard bacterial suspension. Suspension was inoculated on plate containing Muller-Hinton's by swabbing. The antibiotic discs were kept on the plate containing Muller-Hinton agar medium. Degree of sensitivity of antibiotics was determined by measuring the zone of inhibition. The effects of infestation, mainly I did 2. One is on hematology, the other one is histopathology. The blood samples were drawn by severing the dorsal aorta. The hemoglobin content of the blood sample was estimated by using Shallis hemoglobinometer, hematocrit by centrifuging samples directly, microhematology, the blood glucose screen glucose reader, was estimated according to the method of Azevedo et al. 2006. Histopathology has also been done, that a 1 centimeter cube muscle, gill, liver, and intestine of Nile Tilapia are fixed in Boyne's fluid. After 24 hours of fixation, the tissues were washed in 70% alcohol and were then processed in automatic tissue process of dehydration, clearing, and infiltration. The samples were then embedded in melted paraffin bags, trimmed, and sections in microtome at mu thickness. Then sections were taken, and then they were just cleared in alcohol grades. And after that, they were just stained and then mounted, and the stain slates were kept for drying and were examined under a series of microscope, and photographs of the sections were taken using photomicrography. And as far as the results are concerned with regard to water quality characters, actually we had just taken 5 stations, and the parameters recorded include temperature, pH, dissolved oxygen, turbidity, electrical conductivity, TDS, nitrate, and phosphates. And the temperature is, as far as the World Health Organization is concerned, it should be less than 40, but most of the temperature range that is noticed are within the permissible level as far as the fish species is concerned. However, temperature is ranging from 22 to 25. It is suitable for the growth of a fish. And as far as the pH is concerned, it shows variation, and the optimum is 6.5 to 8.5, but in some stations, the pH shows a little higher values. And as far as the DO is concerned, it is 5 to 7 is the optimal range. But again, there is some fluctuation is there, but mostly the oxygen level is also suitable for other fish. And as far as the turbidity is concerned, in all the stations, the turbidity is very high. And that shows that that gives us some problem as far as the fish is concerned. There is a stress factor. And electrical conductivity is also in all the stations, it was above the normal. TDS is also normal. And so also the phosphate. The phosphate is also a higher value. And if that's the case, it clearly shows that the lake is polluted. And as far as the parasites are concerned, we just take a large number of parasites, mainly the branchiurine parasite, the digenean, the cestode, the nematode, and a ANCOSEAFF. And most of the species have been just added. So most of the parasites have been identified up to generic level. And the branchiurine parasites is a Dolaps or a Narum. And as far as the digenean is concerned, Clinostom and Equinostomum. Cestode is a Ligula, Contrasicum. The nematodes, there are 2 species, and a ANCOSEAFF, a single species, was reported from Oriochromis and niloticus. And as far as the parasites of Clarias, Garipinus is concerned, again, the branchiurine parasites is dominating. Dolaps, digenean is Equinostomum. There is an adult parasite is also that's not identified. And as far as the cestode is concerned, there are 2 parasites are there. There is Protocephalus and Bothrocephalus, and nematode is Contrasicum and Cuculinus species. And as far as the bacterial isolates are concerned from the tissues, we just isolated Oriochromis species, adverse species, Pseudomonas, Salmonella species, and Vibrio species. And as far as the prevalence is concerned, Oriochromis 15, then Salmonella 15, and then Vibrio species 15, more or less the same. And Adversella found to be less. And as far as the highest is concerned, Escherichia species is 23%, and followed by Pseudomonas species. And with regard to the incidence, you can just see that the prevalence is, so infestation as far as the length group is concerned, the total, we just collect the number of fish examined is 360, and number of fish infested is 172. And as far as the infestation is concerned, 47.8%. And the total number of parasites collected is 1,573, and the intensity is 9.2. Then with regard to the infestation in relation to sex, it is also seen that the female fishes are more infected than the male fishes. And as far as the intensity is concerned, the male is about 3.5. And as far as the female is concerned, it is 5.7. Then with regard to the prevalence in Clarias, total number of fish examined is 120, and the number infested is 120 again. That means 100%. And the total number of parasites collected is 1,669. The prevalence 100%, and the intensity is 13.9%. Then with regard to the branchiorun parasite, according to the infestation and intensity, the infestation high is 96.6%, and the intensity is low, it is about 2.97. And the Cestode parasite, again, it is much low. The intensity is 1.5, whereas the prevalence is 9.5%. Nematode parasite shows the highest one, and the prevalence is 100%, as far as the intensity is 8.86%. ANCOSEAFF, again, the infestation is high, 59.17, and as far as intensity is concerned, 1.27%. So when compared to, when comparing this one, branchiorun parasites, Cestode, nematodes, and ANCOSEAFF, you can just see that one, it is being dominated by the nematode parasites. It's about 63.69%, and the branchiorun parasite is at 20.61%, followed by the Cestode parasite. With regard to the length group, again, 100% infestation, and the infestation is high in 281 to 320 centimeters, mm, and it's about 48 and then 52. And as far as the intensity is concerned, you can see that 13.91%. We did some work on the hematology in Nile Tilapia, mainly for the uninfected one and infected one. That is with regard to the injection by the Vibrio species. The parameters estimated include hematocrit, hemoglobin, glucose, triglycerides, total serum protein, albumin, and globulin. And the uninfected group values are like this. And as far as the infected group is concerned, you can see that in all the parameters, the values are much less in infected group. That means there is a clear impact as far as the hematology is concerned. And then with regard to the histopathological changes, you can just see that one, the skin, the epidermis is damaged. Dermis is also going to be damaged by that. And apart from that, there is the fibrous nature of the tissue, the fibrous connective tissue. The connective tissue becomes fibrous, and there is an infiltration of the cells, especially with regard to the granuloma cells are infiltrating to a larger extent as far as the skin is concerned. And also you can see that on the gill, the primary gill lamellae, the secondary gill lamellae, you can see that one is quite normal in the uninfected fish. And as far as the infected fish is concerned, you can just see that one, there is a lot of changes that is just taking place, mainly with regard to hyperplastic condition, because mucus secretion is more, and this is just going to give some kind of a change as far as gill is concerned. The lamellae are just going to be fused, and because of this one, there is problem with regard to the respiration, and there is infiltration of the cells, and there is a blood cell damage is there, and that will infiltrate it into the adjacent areas. And also you can see that one with regard to the muscle, quite clear muscle bundle is evident in uninfected this one. But as far as the infected ones are concerned, the connective tissue is just getting damaged, and it turns to be fibrous, and which infiltrates into most of the muscle tissue. And with the result, the intact condition of the muscle is just going to be disturbed. And with regard to histopathological changes in the liver, a lot of changes are just taking place in the liver. And you can see that on the normal hepatocytes are quite intact as far as uninfected is concerned. But with regard to the infected one, you can just see that the sinus and also the blood cells are damaged. Hemosiderin is just going to be developed, which will be just infiltrating into the gill tissue. And the blood capillaries are damaged to a larger extent, and that gives a lot of problem as far as the gill tissues are concerned. And so also the changes in the intestine. And when intestine, the epithelial and the dermal tissues are just going to be damaged, it just reduces the absorbing capacity of the food. And as far as the fish is concerned, this is going to be a real change because the absorbing capacity of the fish is just going to be disturbed when the intestinal tissues are just going to be damaged. And you can just see that external symptoms when you're just going to use this Vibrio infection. And when the experimental setup is just established, and when you're just going to observe according to the days and then the week, you can see that there is development of the symptoms. And you can see that there will be a lot of changes that are just going to take place. And the skin is just going to be lacerated. The mucus secretion is just going to be in excess. And when mucus secretion is going to be abundant, you can see that will attract the secondary infection by bacteria and fungi. And also you can just see that there will be a dropsical condition that is just going to be developed in the abdominal region. And also you can just see that the gill tissues the fin tissue is just going to be damaged. The fin drought is just going to be developed. And once the fin drought is just going to be developed in the caudal fin, you can see that that will progressively just come out. And after some time, the gill tissues and the muscle tissues are going to be damaged to a larger extent. And as far as the internal organs are concerned, you can just see that with regard to gill, there will be pain. And the intestine is also just going to be disturbed because fluid accumulation will be there. And when this kind of a change is just going to be there, the absorbing capacity of or the digestive capacity of the fish is just going to be affected. And these are some of the parasites that we just collected from the Clarias garipinus. And mainly the parasite is with regard to the nematode parasites, and they just affect the internal organ, especially the intestine. Apart from that, they're also abundantly present in the musculature and also the mesenteries. And when you just open the fish, you can see that it is filled with the nematode parasites. And when these nematode parasites are just going to be abundant, and this also just going to cause what we call as the muscle damage. And when you just see this particular fish, when it's just coming to the market, and the whole appearance of the fish is just going to be damaged, and this reduces the marketability of this particular fish. So once if these fishes are just going to be affected, it definitely reduces the marketability and marketability of the fish. And because of this one, there will be great economic loss as far as the fishing community is concerned. So based on the study, what that we just concluded is Lake Hawassa, a closed ecosystem, is under pressure due to the man-induced factors. And that is mainly because of the wetland degradation by cattle grazing and also by the industrial discharges. And many of the physicochemical parameters show values above normal. That clearly shows that the lake is polluted. And this exerts a stress to fish. And fish species that supports the fishery are also subjected to infection by bacterial pathogens and metazoan parasites. And definitely this is just going to reduce the value of the fish. And thus the fish is a real concern as fisheries supports the livelihood of fisher folk of this region. And thus there is a need to protect the lake from further deterioration, especially due to man-induced factors. This is some of the studies that have been just carried out very recently by myself as well as my colleagues in the department. Thank you so much for patiently listening to this talk.